Today what I'm going to talk about is only half of my research project. The other half, maybe for another time. It's on vision and neovascularization. This is a big program project that I'm directing it, and it started from the day that I landed in New York, and it's continued until today. It's concerned, or the major theme of my research are the eicosanoids, or arachidonic acid metabolites. I'm sure many of you know a little bit about lipids, and that most of the fatty acids are not free. They are esterified into phospholipids. You actually need an injury, a stimulus, to release the fatty acid. In this case, we are talking about arachidonic acid. From the phospholipids, once it's released, it's metabolized by many enzymes. And the first enzyme, the first metabolic enzyme that was discovered in the 60s, 50s, 60s was the cyclooxygenase. Today we know that there are two cyclooxygenases, COX-1 and COX-2, leading to the formation of prostaglandin, thromboxane, and prostacyclin. The second metabolic pathway was the lipoxygenase pathway, lipoxygenases, that metabolize arachidonic acid, oxidize arachidonic acid on the different site on the double bond to hydroxyeicosanoic acid, or HEATs, leukotrienes, and lipoxin. My work will be concentrate on the Cytochrome P450. Actually, in '81, this was a very, very new pathway that the pathophysiological significance actually come from Valhalla, from my lab and other faculty members in the Department of Pharmacology. The Cytochrome P450 enzymes, as you know, it's a huge family of enzymes. 100 isoforms, probably 500 isoforms across the board, across species. There are many isoforms of the Cytochrome P450 that can metabolize arachidonic acid to bioactive lipids. Among them are HEAT, hydroxyeicosanoic acid, and epoxyeicosanoic acid, the HEATs. The HEATra is the one that are in the cornea, in the vision system. I will talk today primarily on the HEAT, and the next slide will explain it a little bit more in detail. So again, there are two main branches of reactions, or two types of reactions that the Cytochrome P450s are exerting on arachidonic acid. One is what we call the epoxygenase. The epoxygenases, primarily from the 2C and the 2J family of enzymes, and they epoxide or introduce oxygen onto the double bond, and you get the 4 radioisomers EETs, epoxyeicosanoic acid. These EETs have been identified as the endothelium-derived hyperpolarizing factors. These are endothelium-dependent metabolites. They produce by the endothelium in the vasculature, and they are very potent vasodilators. And they're doing this vasodilation through activation of potassium channels. That's why they're called EDHF. The other branch of arachidonic acid, of P450-derived arachidonic acid metabolism, is done by enzymes that we call them omega and omega-1 hydroxylases. They are primarily of the Cytochrome P450-4A and 4F family. What they do is very simple reaction. They introduce an hydroxyl group into the omega end of the fatty acid, the 20 position of the arachidonic acid or the 19 position. And what you get is a metabolite that is a 20-hydroxy eicosa tetraenoic acid, or 20 heat. This is it. This is the center of my talk. The 20 heat, if it's produced by the vasculature, it's a very potent vasoconstrictor. And when I'm talking potent, this is in physiological concentration. I'm not talking micromolar. I'm talking very low nanomolar to picomolar concentration. It's enough to cause vasoconstriction. It's produced by the vasculature, it will be vasoconstrictor. If it's produced along the kidney nephron, it will inhibit certain channels, including sodium-potassium ATPase, the sodium chloride, sodium-potassium 2 chloride exchanger, and it will, what it will do, it will be a diuretic kind of compound. I will concentrate in my talk on the vasculature, because the vasculature, the bioaction of 28 in the vasculature is what contribute, or the pro-hypertensive feature of 20 heat relate to its formation in the vasculature. My talk is very short, so I'm going to summarize almost 30 years of work in the essence of it in 20 slides. So bear with me, and I will do it. What is interesting about this particular metabolite, the 20 heat, is that it's a metabolite of the microcirculation. It's not formed by the macrocirculation. Blood vessels that are 100 and less than 100 micron in diameter produce this particular eicosanoid. And if we take the microcirculation of the kidney, going from the interlobar, which is about 200 micron, into the arcuate, 200 to 100, to the arcuate, and to the small interlobular that are 40 micron, you get a different bioaction of 20 heat. In the interlobar, it's enhanced the response to constrictor stimuli, like angiotensin II, phenylephrine, etc., endothelin. And in the arcuate and the interlobular, where it is 40 micron, it's regulate the myogenic response, the pressure-dependent vasoconstriction. Now, as you go along the tree, the vascular tree, and you go from the aorta towards the interlobular arteries, from very big vessel to a very small vessel, the synthesis of 20 increase exponentially. So it's really an eicosanoid of the microcirculation. Now, this is a summary again of a couple of years of work looking at the mechanism by which 20 heat constrict smooth muscles. And the way it does it, so it's formed in the smooth muscle cells, and what it does, it inhibits the calcium-activated potassium channel. And by doing that, it's amplify or it's increase intracellular calcium and promote the machinery, the contraction machinery of the smooth muscle. This many years of work, just to mention that the EETs that are produced by the endothelium are in essence a functional antagonist of the 20 heat. 20 heat is a vasoconstrictor, EETs are vasodilators. That's a different subject. What we asked a couple of years ago is whether or not the action of 20 heat, the pro-hypertensive properties of 20 heat, because it's a vasoconstrictor, is restricted to the vascular smooth muscle cells. Does it have any action on the endothelial cells? And the work started a few years ago, and what we found was very interesting. What we found is that actually 20 heat in a concentration of very low nanomolar, here we use 5 nanomolar, this is in cultured vascular endothelial cells. These are human microvessel endothelial cells. Again, I told you the microcirculation, it's the target of 20 heat. And you can see that if you look at nitric oxide production, if you stimulate endothelial cells with ionophore, there is increase in nitric oxide production. This is the way that you stimulate eNOS. In the presence of 20 heat, inhibit the production of 20 heat of nitric oxide. If you put on top of it a compound called 19R, it's 19-hydroxy eicosanoic acid, very similar to 20 heat, but it acts as antagonist to 20 heat. You can abolish the effect of the inhibitory effect of 20 heat of nitric oxide by antagonizing its effect with a very similar product that doesn't have its bioaction but has the capability to antagonize. And probably we're talking about receptor, which is a different subject. So how it does it? How 20 heat inhibit nitric oxide production? And what we found is the following. So what happened to eNOS? eNOS, at the less active, it's anchored to the membrane, and it's by caveolin. And dissociated from HSP90. When agonist is added to the cells and you stimulate or activate eNOS, the activated form is actually a phosphorylated eNOS and two-site phosphorylation, tyrosine 497 and serine 1179, and it gets associated with HSP90. This is the active complex. When you add 20 to the cells, 20 does not affect the phosphorylation state of eNOS. However, 20 dramatically reduce the association between HSP90 and eNOS. By doing that, it's uncoupled eNOS. Once you uncoupled eNOS, 20 production goes down, superoxide production goes up. And that's exactly what we saw. Not only 20 goes down, but superoxide, I will show you in a minute, also goes up. This is just the functional correlate to 20 heat inhibition, to nitric oxide inhibition, showing that if you take these small, microvessels from the kidney, the interlobular artery, we microdissected, and if you relax it with acetylcholine, this is the control. You get a very nice relaxation to acetylcholine. If you add 20 heat, relaxation to acetylcholine diminished. If you add to 20 heat the antagonist, you reverse the effect of 20 heat. The antagonist 19R, not 19S. This is to show you that 20 heat increase superoxide. So it's not only inhibit nitric oxide, it's also increase superoxide. So in reality, increase in the level of 20 heat, increase oxidative stress, reduce vasodilatory capacity, increase vasoconstrictor capacity. So it's actually increase the resistance of the blood vessels, and therefore it's pro-hypertensive. We also find that in endothelial cells, not only it's causing dysfunction of the endothelial cells in terms of nitric oxide action, but it's also activate the endothelial cells. It takes the endothelial cells from a quiescent cell to an inflammatory cells. It's activate, it's release cytokines, inflammatory cytokines, primarily IL-8 MCP1 and IL-13. This happens if you add 20 heat to the cells or if you actually express the enzymes that make 20 heat inside the cells. These are the cytochrome P450, 4A1 or 4A2. In both cases, you get an inflammatory phenotype. And we also showed that 20 heat activates NF-kappa B, which is the de-inflammatory transcription factors, and the MAP ERK1, ERK2 kinase. So in endothelial cells, 20 heat does so many things. What is the common mechanism that we can look and say that it's producing an inflammatory phenotype, it's reduced nitric oxide, and it's produced actually a hypertensive phenotype from the endothelial cells, because not only does the nitric oxide, superoxide inflammation, it also activates the RAS system, the renin-angiotensin system. This, we did a microarray, and with the microarray we found that the only gene that went, the cells were treated with 5 nanomolar, the only gene that was highly induced was ACE, the angiotensin converting enzyme, rendering an increase in angiotensin II levels. So this is another thing that the 20 heat does in the endothelial cells. What is the common mechanism? This is another couple of years' work looking at what is the signal transduction mechanisms of 20 heat in endothelial cells that put together all this effect of 20 heat. We think that we have a receptor. We don't have it in our hand. We are working now on isolating the receptor. What it does, it's all the effect of 20 heat are tyrosine kinase, EGFR, receptor mediated, and MAP kinase mediated, and it goes through the IKK complex, which is upstream of the NF-kappa B activation. And the IKK complex is the one that actually relates to the inhibition or the uncoupling of eNOS, and it's the one that is related to ACE induction. We still don't know how it will go with this. But all this come down to a decrease of nitric oxide, an increase of oxidative stress, an increase in inflammatory cytokines, and you get endothelial activation and endothelial dysfunction. A typical phenotype of endothelial cells in hypertension. So this is in vitro study, and this is actually a schematic representation of what I just said. So 20 heat is produced by the smooth muscle cells. Over there, it's activate the contractile machinery, and it's act on the endothelial cells. Endothelial cells do not produce 20 heat. It's act on the endothelial cells, perhaps through a receptors, activating the cells, producing endothelial activation, endothelial dysfunction, and vasoconstriction of the blood vessels. Overall, it's conducive to increased vascular resistance and therefore hypertension. And this is again a summary of a lot of years of work where we actually identify models of animal models that actually show this type of biochemistry, which means they show an increase in the synthesis of 20 heat in the vasculature, and this is the basis for their hypertension. Because these are the models. If you do the androgen-induced hypertension, if you just inject androgen to rats or mice, they become hypertensive. DOCA salt, the SHR, the spontaneously hypertensive rats. If you increase if you express the cytochrome P450 that make 20 heat by viral vectors, either general or specifically into the vasculature. We have some transgenic mice that express high level of the cytochrome P450 that makes 20 heat. All these animal models show one common biochemical event, and this is an increase in the synthesis of 20 heat in the vasculature. And you can show that the hypertension is dependent on this 20 heat by using antisense oligonucleotides or siRNA against the vascular P450s. You can use inhibitors of the production of 20 heat. You can use antagonists to the 20 heat bioaction, and all this can inhibit the increase in vascular reactivity, inhibit the increase in peripheral resistance, and inhibit the increase in blood pressure. All these models are 20 heat dependent hypertension. And this is just the last 2 years of work showing the relationship between the 20 heat pathway and the RAS, the renin-angiotensin system. In this particular project, what we did, we developed lentiviral vectors for vascular endothelial-specific expression of the 4A2. This slide just shows the specificity or the tissue-specific expression of this enzyme. Either we did it in vitro, in vivo, showing the colocalization of the P450 with the CD31 endothelial-specific markers. In this model, again, you have increased synthesis of 20 heat. So we had 4 groups of rats. One group received the control vector. One group received the 4A2, the CYP4A2. One group received the CYP4A2 plus the inhibitor of 20 heat synthesis, and another group, the control group, with the inhibitor of 20 heat synthesis. You can see that the vectors, the 4A2 vectors, increase synthesis of 20 heat. With the inhibitor, you inhibit the synthesis, and the blood pressure is a mirror image of the 20 heat synthesis in the vasculature. In the same model, we could show endothelial dysfunction, which is evidenced by reduced relaxation to acetylcholine. You see the EC50. You have also a reduction in the expression of enos and phosphoenos, so you have a reduced capacity to make nitric oxide. These particular mice are suffering from oxidative stress, they are hypertensive, and they have renal damage as well. But if you take these mice, so you give the virus, this is bolus injection of the virus. You give the virus at day 0, 1 time. Blood pressure goes up within, actually it goes up within 5 days. It goes up, stay up, 20, 30, even 40 millimeter mercury in some rats. And at that point, we give losartan, angiotensin II receptor 1 antagonist, losartan. And blood pressure comes down. You stop the losartan and blood pressure comes up. Actually, we have a couple of experiments. I don't have the slides here. You do the same with a 20 heat antagonist. Blood pressure comes down. You stop the antagonist. Blood pressure comes up. So it's, we can reverse it either by losartan or by 20 heat antagonist. In this model, we can show very nicely that there is increased angiotensin II, and there is increased expression of ACE. It's all dependent on 20 heat because you can reverse the effect by using the inhibitor, the HET16, the inhibitor of 20 heat synthesis. You reduce plasma angiotensin II, you reduce vascular tissue angiotensin II, and you reduce ACE as well, expression of ACE. So this was the animal model. Is there is any correlate to human? Yes, there is. And we're just starting now to work with human materials. This is a big project that we took on ourselves. This is actually collaborators from Australia that did a correlation in hypertensive individual. They look at oxidative stress and 20 heat excretion, as well as endothelial function by using the flow dilation in the brachial artery. And you can see that there is inverse relationship between the level of 20 heat in the urine and the changes in the dilation in hypertensive individual. And you have a direct correlation between oxidative stress, here is isoprostan, or glutathione, gamma-glutathione transferase, and 20 heat level in the urine. And there are also a group in China that shows that there is a polymorphism of the 4F2. The cytochrome 4F2 is the one that in human localized primarily in the vasculature. And there is a polymorphism that actually render this cytochrome P450 more active and make more 20 heat. And this polymorphism is associated with increased urinary 20 heat synthesis and with hypertension in humans. So there is a human correlate to whatever we're doing in the lab. And this is, again, just to summarize. And that's it.